Good evening, sir.
Hello, good evening, Dr. Nandini.
Good
Yes,
evening.
ma'am.
Good evening, ma'am. I'm sorry today I was late. From tomorrow, I'll take care.
No problem. I thought so. You must be in some emergency, so I put a message. Okay. Yeah. So, all right. So, first of all, good evening, and I will introduce myself first. And just a second. Just keep it on mute. Yeah. Good evening, Dr. Nandini. I'm Sharvari Kulkarni, and I have for 14 years of experience. I completed in computational biology. My area of specialization are all the tools of basics and advanced of bioinformatics, practical and molecular docking, and MD simulations on all the platforms and operating systems, network pharmacology, and next generation sequencing, in which I have my international publications also. I'm working as a chief technical officer for BDG Life Sciences, and I am in the last stage of PhD in computational biology. So this is my brief introduction, and I would like to just know your brief introduction so that I understand what is your background. If you are very new to bioinformatics or you are from the life sciences, I just want to know that.
Yeah. Hello, Sharvani ji. This is Dr. Nandini. I am actually a veterinary doctor, and my core subjects were BVSC and then MVSC in animal genetics and breeding, followed by PhD in animal genetics and breeding, right? And
Uh-huh.
then, and then I joined here as assistant professor, come junior scientist in the Department of Animal Genetics and Breeding in Birsa Agricultural University, and I have been working on the same designation for about 10.5 years. So over a
Super.
decade. Okay.
Nice.
Yeah. And we have this field actually, other than animal genetics and breeding or molecular genetics, we have to work on bioinformatics. That is it. And I have taken online courses also, although honestly, I'm not a pro in big data analysis, but I have basic knowledge. Like molecular docking, I first learned at Tanuvas through offline course, but it was a very brief introductory course about docking in 2013. And then after that, I did a couple of online courses on bioinformatics.
Mhm.
So, yeah, I'm trying to learn because I know without good command on bioinformatics, my analytical part lacks on important aspect.
Okay.
So, yeah, but I need, kindly treat me as a layman when you teach things.
I wanted to ask you that only. Actually, the purpose of asking in the first session is that only. Do I need to really start from the scratch? Or sometimes people have done something, then I don't go into much basics. But if you frankly tell me that I will treat you as a beginner, I would prefer that.
Yeah, kindly. That would be better. And
Thank you.
ma'am, could you please, if possible, could you please give me an introductory part, like what all were you going to cover, in this training?
Okay, so
Hmm.
first day, the plan is like we have it as a 7 days workshop, right? So the first we will start, this is very basic and advanced combination. So I have separate courses which goes about this. So first this course, then generally people come for docking. I teach docking very extensively on all the platforms and with around all the specialized tools and batch simple docking, multi-docking, protein, nucleic acid, everything. Then I cover MD simulations also, but those are different workshops and those are extensive, like 7 days, 10 days, like that. This is for the beginners who have first exposure to bioinformatics. So once you understand these basic tools, you get good hands on operating these tools. Then if your interest still persists, you can definitely join the other workshops. So in these 7 days, we have divided each session into 2 hours, and every day we will be doing first the understanding of the application, some theory of it. Wherever required, I will also teach you algorithm. Then I will give you demo. Then I'll give you time and task to perform. Now, generally, when there are many participants, I tell them to share the results on the WhatsApp group. Then I validate, check each result. We discuss the results, and every session will be followed by question answer. Here, you are the only participant, so we can have one-to-one interaction also. Anyways, even if there are 20-30 participants, then also most important part that is the benefit of my workshop is one-to-one interaction. You are free to ask any simple and complicated doubt, but I would appreciate if you ask at the end of the description or explanation is over. Now in our case, we can also do the screen sharing if you want. If anywhere while performing you are stuck, I will give you access, you can share the screen. Otherwise, if you are performing, you are stuck, you can take MIC and talk to me. So today we will start with very basic, right from what is bioinformatics, some history, not much. What are the milestones? Then what are the applications, where exactly in all the domains bioinformatics stands. Then we will spend some time on that slide. Then we will be focusing on the information of the databases, what are the classifications, and which three main databases we are going to commonly use in our studies, basic, advanced, everything. So we will first learn about them, then we will perform hands-on exercise on each database, how to retrieve the files. Then I will give you time to do that. I will check if we have taken correct files. And then today, if this goes fine with our time schedule, then today I will also cover primary structure analysis tools, which are ProtScale, ProtParam, and uh, TMHMM. After that, tomorrow we will do step by step, we will gradually move. So tomorrow I will plan to take secondary structure prediction with the Chow-Fastman server, and we will do the exercise for Swiss PDB viewer also, and visualization of the protein by Rasmol Pymol. So after that, we will be doing interaction studies, which is again, this protein structure visualization is very important when you are pursuing bioinformatics. You have to get good hands on this visualization software. So that will be our next objective. After that, I will focus on protein interaction studies, which are also very important when we are discussing any pathways in your study or the domain. Also, when you are talking about the genetics, animal genetics, there are pathways studies which are important to understand the physiology path and pathology. So these proteins which are interacting with each other is studied with the help of tool known as streams. Then we will do uh, blast analysis, which is again a very important study where you compare the sequences of different species in order to check their similarity or homology. Then we can do pairwise sequence alignment. We will also learn multiple sequence alignment. We will learn primer designing. Then we will do phylogenetic analysis study, how to interpret it, how to construct the tree. What is the main important application of it? It is to understand the evolution. So it will be helpful in your veterinary study also. So it is like learning everything, and you will have to decide on the basis of your objective how to implement it in your domain. So it is such a multidisciplinary field. We all the researchers use it for their different purposes. For example, as I told you, my expertise is computer aided drug designing. So I will be more focused to use the tools of docking, MD, I'm a CADD expert. You are doing genetics, so you might be using more tools which are related to genome browsing. That also I'm going to cover. We will cover two genome browsers, NCBI and UCSC genome browsing, which are extensive to understand the genes and genomes, complete genetic study. Then we will do structure prediction by FIRE2, which gives us the sequence profile and mutation profile studies, which are significant and non-significant mutations. And last day, we will do the allosteric regulation or allosigma server study. So this is the holistic plan. We'll do step by step. In between, if you have any queries, you can ask. My pattern of teaching, as I explained, first will be the explanation. Today we will have more of the discussion, and we will try to do some basic things which must be easy for you if you have done online courses before. But no worries, even if you have forgotten, it's no issue at all, because as I told you, I'm going to teach you as a beginner student. So first, let me share the slides. Each day I will be performing the task in front of you, giving you the time. Apart from that, I will be every day during the session sharing the document of the steps with you. So that will be extremely useful. You just have to follow the steps one by one like a manual and perform the task. At the end, I will check your results.
Can I say, ma'am, a few things?
Huh? Huh? Huh?
The thing is that all the topics definitely I need, and because that's why I have enrolled in the course. But as
Hmm.
you could rightly guess, I mean, the primer designing, phylogenetic analysis, BLAST,
Uh-huh. Uh-huh.
genome, part, they are all really very important for me. And
Yeah.
I don't have honestly much knowledge about any of these. So I want to really, I'm looking forward to learning. And of course, molecular docking fascinates me like anything. So
Yeah, yeah. It is fascinating, definitely.
yes, ma'am. So I'm really looking forward to it, sir, ma'am.
Okay,
Okay.
so but then Nandini, docking is a separate workshop, so you can join that afterwards, after this.
Acha, I thought in this you will be focusing on docking. So,
No, no, no. It is only you. If you see the topics, it is only basics and advanced.
it's
Docking
good.
is,
It's that also is good, ma'am. Separately, if it is there, I will that.
yeah.
Good.
And I vouch you, it is my top ranking workshop since 6 years. I get 100% feedback. This is not for boasting, but it is to tell you that I teach thoroughly the docking. Means at the seventh day or eighth day of the docking, you should be docking. You will be docking expert, because
Wow.
I love that topic so passionately that I'm totally now engrossed in it. So I love teaching that. You can join. I advise this to those people only who show really good interest and genuine interest. So after you learn these basic things, I will inform you when the next docking workshop comes. Definitely I'll help you to become docking expert.
Thank you, ma'am. That means a lot. Okay.
Yeah, my pleasure. And after that, if you are still fascinated, you can pursue MD simulation also. So it
Okay.
goes that way. How well you are doing, that will definitely clear your objective, right? Okay. Yeah. So first thing that I would like to start with is a very basic discussion about what is bioinformatics and how did it gain importance. So as I told you, I'm into this field since more than a decade, 15 years. Yes. But when in that time I was teaching as a professor like you for engineering college, that time bioinformatics was only limited to the books, foreign author books, that's all. But actual practical application was not much started.
Yes,
So,
ma'am. Yes,
yes,
ma'am.
right. So in India specifically, we used to only understand and mug it up and get the exam clear. That's all. But after that, it started gaining importance. So I can definitely see its drastic change post COVID. Till COVID, it was not much in the focus. People were not much aware of using all these softwares. But after the COVID strike, researchers started understanding that even if you understand how bad the crisis was, labs were getting shut. But at the same time, people started understanding the importance of shifting this research approach from the wet lab to computers. And this is exactly the time where it was, I can say, game changer for a computational biology or bioinformatics. So first corona vaccine was also entire bioinformatics work, which was done by Moderna company on RNA-seq. So it was one first RNA sequenced vaccine. Thereafter, people started understanding the importance of computational biology. This picture I have purposely kept on the screen to make you understand how the data generation occurs in bioinformatics. So it starts here in 2000 and it rapidly increases. We are somewhere here now. In 2025, we are expecting about 40 to 60 gigabytes of the data to be generated. That still, if you expect 2030, it will reach to 70 gigabytes of data. It is huge amount of data. So you should think if the speed and the data generation amount is so huge, if you are not able to organize this data in an efficient manner and in a proper manner in order to retrieve it properly, what is the sense, right? Doesn't make any sense. So you have to design a proper efficient system to use this data in a proper and sensible manner, and that is where exactly the role of bioinformatics come into picture. So in bioinformatics you are storing, organizing and retrieving the data in order to implement some important practical implications or interpretations. So wet lab and dry lab, they go hand in hand. You take a data from wet lab, process it, and you again give interpretations to the wet lab from the dry lab. So both of them are interdependent. Now, this field, as I mentioned, is not a very new field. It has its roots in Human Genome Project, which initiated in 1988 and declared completed to be 2003. Major goals in front of us were to determine 3 into 10 raised to 9 base pairs, identify approximately 30,000 genes, and computational tasks in front of us were storage, indexing, building contigs, scanning for genes. This is the draft of finished and unfinished draft of Human Genome Project. These are few milestones in bioinformatics. So like we first started with Watson and Crick DNA model, NW means Needleman Punch algorithm, PDB. These are some important landmarks, I would say. BLAST, FASTA, SwissProt. So this was to build your background. Now I would like to start with the first point, introduction to bioinformatics. Then we will start our discussion with databases. And then today we will perform three tasks, prot scale, TMHMM and prot param. So let us discuss some conventional definitions of bioinformatics. It is the computational branch of molecular biology. It is a multidisciplinary branch with vast applications. Why I'm saying it is a multidisciplinary branch? Because it is related to many disciplines and with vast applications. I will focus on one slide to explain this point more elaborately. Going ahead, I would like to define bioinformatics as conceptualizing biology in terms of molecules and then applying informatic techniques to organize the information associated with these molecules on large scale. It works as a sequence, 3D structure and function. So talking about this point, we are using nothing different, but we are using the concepts of molecular biology, and we are also taking at the same time help of information technology, which are advanced computer techniques, in order to come up with the speedy conclusions and accurate conclusions related to this study. Talking about its multidisciplinary approach, I would like to focus on this slide more. So at the center, you can see this bioinformatics. Now I have splitted it in different branches. So one branch is protein sequence analysis. Then you have this DNA sequence analysis. In the protein sequence analysis, we call this in a more technical or accurate term as proteomics. So in the protein sequence analysis, we do primary protein analysis and structure analysis. That means properties of the protein. So in that, we will be doing today prot scale, prot param, TMHMM. When you are searching for the properties of the protein is proteomics. In that we will cover tomorrow secondary structure prediction also. Then comes the little bit part of the genomics, where you are doing sequence analysis like BLAST. I will be teaching you both the exercises of the BLAST for protein as well as nucleotide. Then after that, we will be doing primer designing. So in the genomics, primer designing part also comes. Then we will be doing, here itself, we will be doing multiple and pairwise sequence alignment. Then phylogenetic analysis. Phylogenetic analysis is to understand the difference between the same protein in different species and put them in the evolutionary order. So this is also very important thing. For this, I will be teaching you one online server, Cluster Omega, and one Mega X, which is the latest one. After that, as I mentioned, this and more, many more softwares, like I mentioned, browser, genome browsing, AlloSigma, FiRay2, InterProScan, all of them. This will be total part of this 10 days, 7 days workshop. Apart from that, this molecular interactions is one part of the bioinformatics, which is focused on the protein docking, protein nucleic acid docking, and protein. There are some specific research paper case studies also, which I discuss with the specific uncommon types of docking which are done nowadays in research. So that considers that is comprised of 8 days workshop. Then molecular modeling I teach. Now I have clubbed it in the docking itself because it is again very important. So I teach it by AlphaFold 3. If the PDB structure of your protein is not available, then PDB structure of the protein is not available, then how to build your model. So that also I have incorporated in the 10 days docking workshop. Then this is MD simulations. After doing all this, you can go to one step above for MD simulation. It is again a 10 days workshop where I teach protein by GROMACS, protein by NMD, protein and ligand complex by GROMACS also. So get expert in simulation, because everything I make each and every participant do on their own systems by setting up the system, and I see to it that everyone gets their results. They make their own files, write their own commands, programs, everything. After that, there is one drug designing part, which is like starting from target identification, validation, lead identification, optimization, and admit. So that is again a different workshop, but it is just actually speaking, drug designing is together docking and MD. So entire drug designing pipeline will be first consisting of molecular docking. When your complexes are past the test of docking, then only it is eligible to go for MD simulation. Once it is thorough with MD simulation, it will go for final admit analysis, absorption, distribution, metabolism, excretion, and toxicity analysis. And then only it can be launched in the like market for the approval, FDA approval, ND approval, all that. So this test passing of the drug from the first to last step is very, very important. And that is like a boon given by bioinformatics to perform this computer aided drug designing. All right. So now I have made one simple image to show you what exactly we are doing in bioinformatics. You must have heard different terminologies for bioinformatics. It is called as a virtual lab also. Why it is called as a virtual lab? Because virtual is something which you are creating. It is not present. So from the living cell information, we are creating replica of living cell in the form of virtual cell. How do we create it practically? We are providing information of sequence, biological knowledge in the form of sequence, in the form of structure, and we are making a virtual cell. And when you give all these data with the help of different programs and softwares, you are coming up with principles and practical applications. So this was for you to get an understanding of the bioinformatics. Now, I will tell you one real world example. What kind of projects commonly you do in bioinformatics? So when I'm talking about my field, when I get projects on computer aided drug designing, how do we work on that? So suppose you know this fact, and this is exactly from where the importance of bioinformatics or CADD started increasing. So when I started working on COVID, when there was a pandemic, I started with COVID. You know, COVID has a receptor protein like membrane and spike. Those have specific PDB codes. So first we take this PDB file of the protein, and suppose we know that curcumin, we have heard that time also that turmeric or curcumin family has good inhibitors which can inhibit good drugs which can inhibit COVID infection. So for suppose one client comes to with the data of COVID inhibitors like curcumin data in the form of SDF files. These SDF files you can take from any database like Impact, Drug Bank, PubChem. What I do in the first step is I will take the COVID protein receptor, which we call as a target, dock it against, suppose I have 500 ligands, so I work on 1 lakh ligands also at a time. So the technology is so advanced. I do batch docking on Linux system, though it does not support on Windows. 1 lakh molecule data also you can handle. So see, for the simplicity and understanding, let us say I have 500 molecules. So I dock it with the receptor protein on the basis of binding affinity or docking score. With the help of Discovery Studio visualization, PyMol visualization, interaction study will be done, and you shortlist only suppose 10 ligands out of that. These 10 ligands are my best identified dock complexes. But still, you can't say guarantee or you cannot promise that they are going to be good inhibitors. So what is the next level of validation? Before 5, 6, 7 years, it was okay that you give a docking data and your paper will be published. That is fine. But now I can see your study is not complete for computer aided drug designing without giving a data of MD. So out of these 10, all you can take for MD simulations for time step of 100 or 200, depending upon your objective. on the Gromax or Desmond software. After that, you get a data of stability. So docking, I would say, is the identification of the stable complexes, and MD is the validation. After getting the data of simulation, out of these 10, only 2 to 3 will be passed. Then you do now latest techniques after AI and ML also they have increased. After simulation, some people are now doing MMPBSA or energy contribution studies, DFT studies. So like Nandini, I will tell you, if you want to explore, it's limitless how much you want to explore and learn. But now we are going to gradually become experts. So let us start with the basics. Now, I am going to talk about the database. In general, how do you define the database? Database is any type of collection of information. So when I'm saying biological database, it is a library of life sciences information collected from scientific experiments, published literature of high throughput experimental technology and computational analysis. So they are broadly classified as sequence and structure. As the name itself says, sequence means it will be storing the sequences and structural means it will be storing the structures. On the basis of the source, they are also classified as primary and secondary. Primary databases will be only the experimental information base. That means your X-ray crystallography, NMR data will be stored in the primary databases. So PDB, NCBI, all are example of primary. Secondary databases are where the information from the primary is processed and treated or filtered, then it becomes your secondary. So like you have many secondary databases like TREMBL is there, Prince is there, Smart is there. So when the databases were introduced, we were having only three major databases. But today, based on the scientist requirement, thus amount of study you are contributing, we have separated the databases according to the type of information and the objective. So today we have minimum 1500 databases. So fungal, plant, viral, SNP database, Indel database, everything is there. Okay. Now, first, I'm going to focus mainly on three databases, NCBI, Uniprot or Swissprot, and PDB. So the first database that we will see is NCBI. This is the home page of NCBI. We will take one query. Query means the question which you are giving to the database. Let us say I'm taking insulin Homo sapiens. Then we will do PDB and then we will do Uniprot. Then if this is completed, then we will move to prot scale analysis. Now I'll simply show you how to navigate through NCBI. First, I will advise you to do properly everything in the workshop and to record everything as results. What you should do is always
Ma'am,
make.
ma'am,
Hmm?
just one, sorry. Will I get the recording? I mean, so that I
Yes,
can
yes, dear.
revise
Yes,
before
yes,
next
dear.
day.
Absolutely, absolutely.
Thank you. Thank you so much,
After
ma'am.
every session, it is actually automatically loaded on our computer's cloud system of Zoom. So when it is completed, technical team will give it to you. No problem.
Thank you, ma'am. Thank you, ma'am. That way I will be able to revise because I told you, na, I feel like a layman still.
No, no,
I mean,
no, don't worry. I will give you all material. Do not feel any hesitation to ask also. Everything you will get in the form of notes, recordings. Don't worry.
that's so nice of you. Thank you, ma'am.
Yeah, my pleasure. Okay, so we were about to see how to navigate smartly through the databases. So the first very important means if you say that you have done some computational biology and bioinformatics, people will definitely expect that you have done NCBI. So you will go to NCBI. Simply click on NCBI. Here you can see NCBI. I will not say it is a database, it is like a huge consortium. It has all the data in it, whichever you want to access. And the best part is there are few tools also which are linked to NCBI. So if you see this drop down, you have lot of information which is properly segregated. So it is so well sorted and organized. If you want to see only the gene information, you can select gene. You can select filters over here. So, and we are going to learn in the process of the workshop, the softwares also which are running on NCBI. Like blast we will do, which is based on NCBI. We will do primer designing, which is based on NCBI, and we will do browsing, genome browsing, which is based on NCBI. Now I just want you to show the basic how you take the FASTA sequence of a protein or nucleotide from NCBI. So for that, let us say I'm selecting here protein. You can type any protein of your choice. Not required that whatever I am doing, you select only that as a query. Whichever protein you are working on, you are interested, you can practice for that. So I can go to Homo sapien protein sequence from here. Always relevant first species. So otherwise, I'll show you this kind of table you get. I am not given the name of the protein. So to be more specific, suppose I give here you Homo sapiens insulin. Yeah, so we get insulin. I will try to go for a complete protein and not the precursor or partial. So I click over here, insulin, Homo sapiens. Yeah, so this is one NCBI file. And one NCBI file is so well organized and informative that it is like reading 10 to 15 research papers on that topic. It will be gathering all the information. So this is how it looks like, a typical NCBI file. It starts with your header. It is giving you then the GenBank. GenBank is another database which has all the nucleotide sequences saved for that particular protein or gene. Locus is the location in the database. This is the length. This is the date of deposition, accession number, version number. Version number will change if someone adds some information and update it. Then comes the lineage or a taxonomy. Then the journal in which this information is present. Signal peptide, mat peptide, protein sequence, CDS sequence, everything is there. At the last, you will see the amino acid sequence. Right now, what we are interested in is to learn how to take the FASTA sequence of this. This is very important. From tomorrow, I will be telling you directly in different tools, you will have to copy and paste your FASTA sequence. So you should be very easy and fast in that. So see here in this legions, I will click on FASTA. Yeah. So once you click on the FASTA, you'll be able to see here this kind of sequence. So the FASTA sequence always starts with the greater than symbol. So in bioinformatics tools, you can't expect that only any format of the sequence will be accepted. No, it will not be accepted. There are always stringent formats of everything with the symbols. When I say FASTA file, you have to be sure that it starts with greater than symbol. So it starts with greater than symbol, accession number, name of the protein, and then it is followed by the amino acid sequence. This is one format which is universally accepted. There is another format known as PIR. It is used sometimes in software where this particular entry is written at the end with star. So there are different ways of databases, ways of the file formats, how they are written. Most commonly used is FASTA. So we are going to focus on that. So when I tell you to take the FASTA sequence, you are going to come over here. You are simply going to copy and paste the FASTA file. So for our practice today, I will advise you to do one exercise and show me the download of FASTA of any protein of your choice from NCBI. But let me explain all three databases together, then you can do the task. Now, the next one I will do is UniProt. UniProt is again a protein sequence database which is coming from UniProt KB, and it will be divided. It is divided into two, SwissProt and TRMBL. So SwissProt is the manually curated data by scientists, and TRMBL is the translated or computer annotated. We will be using SwissProt. Like here, you just have to give your protein's name. And we will be mostly focusing on UniProt sequence retrieval. The reason is it is manually annotated database. So the entries are filtered and the sequences can be retrieved very quickly. It is not like NCBI. NCBI you must have seen. It is like a Google. You get so much information and then you have to find your relevant one. It is not the case in UniProt. It is very well organized in the form of table. So suppose I click over here, insulin is already present. Suppose I click on insulin. Search. Yeah. So this kind of information you get. Now out of this, it's so easy to select your desired one. I will go for Homo sapiens. So this one, again, this is one more concept that you should be knowing. This is called as an accession number, this P01308. Accession number is like a roll number unit class. Okay, so how do we have our identity as a roll number? Similarly, every protein or nucleotide will be having its entry or accession number. For UniProt, for every database, it is unique. So for UniProt, it starts with this P-O and followed by numbers. So let us click on this. So this is a typical UniProt file. It has other features also, like it will tell you the function, gene ontology, the pathways, very informative. Okay. But right now, all these databases, dear, you will have to also, if you want to become expert, it's up to you how much you want to explore. I always tell this example to my participants sitting in the same workshop. One person can do a task in half an hour. Another person who want to go into depth because it is so informative, another person can spend 1 week also on the same software. It's up to you and your urge and enthusiasm of learning. So now what is our interest here is we are now learning to download the FASTA. So I click on this download legend. It will ask you the format. So I will click on the FASTA canonical. Do not get confused between any other format. We will go to FASTA canonical and click on download button. So same sequence, did you observe? Same sequence which I had shown you in the NCBI will come here. So It's your choice from where to take the protein FASTA sequence. Again, you see it is following the same pattern. Greater than symbol is very important. Then the accession number, then the species description, and then the actual sequence. So this is the second database. Now the last one which I want to show you is PDB. PDB is excellent reservoir of the protein. What is the difference? Major difference between PDB and UniProt and NCBI is PDB stores structural information, which is very important because when you are doing MD docking, it is not sufficient to give only the data in the form of sequence. Very, very important is how they are arranged in 3D space to work on that. So PDB file is very important input file every time. So in the PDB, you have to first learn how to take the PDB file. Let us say I can do it for any random protein. Suppose I'm doing it for SARS-CoV. So you can select any protein. Suppose I'm selecting this one. So this, it will come with these codes. First one is almost every time first code or first file is most relevant. So we will click on this. Then here again the same thing. You have a lot of information in the PDB file. The date or the object entry, PDB code, the resolution of the experiment, experimental data, the literature which supports the work on this structural analysis, what are the entities in that, which protein is involved, if there are some other parts than protein like metal or anything, that also is included. Right now, suppose here, like zinc is present, so that is also mentioned. And you can see the interaction also between the zinc and protein. So all this is very interesting. Right now, what we are interested is to download the PDB file. So I go to this download and go to this legacy PDB format and click on this legacy PDB format and download it. Now, all these files you will transfer to you can make a folder, you can give it name like Bioinformatics Workshop, so that all the input files, output files, results files you will maintain in one folder. In future also, after 2-3 months also, if we refer to that, it will be clear. All the notes, everything you just keep it in the one folder. Now, your task will be first, Nandini, you will go to NCBI, take any protein of your choice, copy paste its FASTA sequence in a blank notepad, save that into your working directory. Then you will go to UniProt, take any sequence of your choice, copy paste the FASTA sequence and save the file. You will name first WordPad or Notepad as NCBI FASTA and second as UniProt FASTA. And third, you will simply download PDB, any protein of your choice, transfer it, just keep it as PDB and don't try to open it because you can't open PDB file just like that, like a Word file. It needs supporting software, which we will be seeing tomorrow. Like I will teach you PyMod, Rasmol, Swiss PDBware, in which you can open your PDB file. PDB file can either be opened with the help of command prompt or it can be opened with the help of visualization software. Okay, everything is clear till this part, Nandini?
Yes, ma'am.
Very good. So will you do this task now in front of me?
What did you say last one, ma'am?
No, you just have to take these 3 files so that I understand you are able to follow everything and you are good in that. You have to do these 3. First, FASTA sequence from NCBI, second FASTA sequence from UniProt, and third PDB file. All these you download and save it into your bioinformatics folder. Easy?
Okay, ma'am.
Ha. So would you like to share your screen or you will send the pictures? What is most comfortable to you?
I will share the screen. Can you please give me just 5 minutes, ma'am? Actually, just 5 minutes. Okay, ma'am.
Yeah, yeah. Okay, okay, okay. No problem.
Hmm. Ma'am, actually, hello, ma'am.
Ha ha, Nandini.
Actually, today my husband came late, and when at the same time he came when so I was late also when your class started. So I was
Acha.
having dinner and listening to your class. So
Oh,
so I have from tomorrow onwards I will be cautious. I will try to finish I mean, although few things are unavoidable, you understand. So
Yeah, I understand as a family person, no problem.
that's why my laptop was not open, and I'll
Huh.
keep open from tomorrow itself. Tomorrow.
Yeah, yeah.
Although in online classes, I do keep it open. It was
Uh-huh.
my fault.
No
Actually,
problem.
today I came from Hyderabad. I had gone
Acha.
for some one training in Center for Cell and Molecular Biology.
Haha. Okay, okay.
It is
You
a
had gone
CSI.
there for conference
No, ma'am, I went for a training on cell
training.
and tissue culture.
Acha, great, great. So you
It
came
was
back from where are you residing now?
I'm in Ranchi, ma'am. Ranchi,
Ranchi.
Jharkhand.
Okay, so you traveled from Hyderabad. Okay.
Yes, ma'am.
Then
Yes.
it must be hectic also.
Absolutely, ma'am. Today morning only I came.
Huh.
And
You had
and
to work
then
today?
ma'am, and then I went to office. Today morning I
Oh
came
my
at
God.
about 12. I reached home. I just took bath and had a breakfast and then rushed to office. And then
Oh
I came back and then I had to, I mean, did some of the household chores, kept the clothes for laundry and all. And then I remembered, oh my God, ma'am, I have to
I
attend class. And I was super excited for the class. Honestly, I wanted to attend.
but I appreciate your enthusiasm, and I always appreciate this kind of I mean the woman who try to manage everything. I'm also one of them. So
I
my
understand, ma'am.
today same thing. I was like in big struggle and exhausted. Now that we are talking, I'm sharing. Otherwise, I'm very
I
professional.
understand, ma'am. It's
My
okay.
daughter's my daughter's maths paper tomorrow. So whole day
Oh,
whole day it was like a brain trauma for me. So, but then we have to work. There is no excuse. So,
absolutely, absolutely.
I am
This
feeling good. At least the participant when is good, then you feel good about the work.
is this is
No
actually the
problem.
same.
Then, Nandani, so what I will advise, if you are tired, it's okay. We will manage. We will stop today. You take a rest.
No,
Then,
no, ma'am. Then, I mean, towards the end, I will have to rush, and towards the end, I want to keep time to ask you doubts as well. I
okay.
understand, ma'am. You are also busy. Uh,
Yes. So, what we shall do now, we will perform hands on.
yeah. Now I have opened,
Okay.
ma'am, opened the
Okay.
computer.
Fine.
So I'm
Fine.
opening LCBI. And yes, I have gone through a couple or more of online training, but they were
Mm-hmm.
not the way you taught from the scratch. Maybe that I missed in all those training. So basic
Haha.
things like I was lacking. So today, the way you were teaching everything, it was really good. I appreciate it. It's not that you must have got good rating. I now I accept the fact. So
Yeah, yeah, I do get lot of good feedbacks
Mehnat,
and
the
ratings
labor you
because
put is visible.
I know, even I know my patience level is very good, and I teach with love and from the beginning. Because see, many
Absolutely.
people, you are a doctor, you are a PhD, but many people are not very comfortable with computer at once, no? So you should understand that and give them a time to be comfortable. That's what I believe. Okay,
Yes, ma'am.
so no worries. Though you share the screen, we will do it together on your screen.
Okay, that will be good, ma'am. I'm sharing it and I'm opening the
Yeah.
link in my
First, let
laptop
me,
only.
I think I'll have to give you host access then only.
You'll have to give me?
Host access. Now you can share. Now you are the host, so you can share the screen.
Ah, okay, ma'am. Okay. I'm
Yeah.
clicking on the link to join from my computers for that.
Yeah.
Okay.
Yeah. Yes.
Okay. See, I'm trying to join, ma'am. Kindly accept. And then I will leave this mobile then. Leave the access via mobile.
Yeah, but I can see you in the waiting room. To admit, I cannot see you in the
Yes,
waiting room
ma'am.
yet.
Oh, you can't see me. Okay.
Hmm.
Wait, ma'am. Escape. Yes, the actually you made me host just now. I'm waiting, ma'am. You have to admit me.
I
Huh?
can't see you waiting.
Huh. You can't see me, ma'am. Okay.
Yeah, I cannot see you in the window to admit.
Oh, we have let them know you are here.
Hmm.
Huh. It is telling we have let them know that you are
But then
here.
why can't I see?
Should I rejoin again? Or maybe because you made me the host?
Maybe. So I'll just make myself host again and then
Yes, yes,
let's see
ma'am.
if I can.
Yes,
Ha, now
ma'am.
I can see. Perfect. Yeah.
Okay, okay. Huh.
Now you have, right? Ha.
I'm leaving from this. Please wait,
Ha
ma'am.
ha. True.
Yes.
Now I will make you host on this.
Okay, ma'am. So this is my screen, ma'am.
Yeah, I can see.
Mhm.
Yeah. So first go to NCBI. First make the folder on the desktop. Name it as Bioinformatics Workshop. And every time make a practice of saving all the results, everything into that.
Okay, ma'am.
Because this is my usual experience with the participants, they do learn very nicely, they perform, but since we all are so professional and busy people, after the workshop is over you get into your work, but after 2 3 months you realize that you have lost the data. So better to make one confined folder so that it is helpful for you to revisit it, right?
Okay, ma'am.
Mhm.
Mhm.
I can't see your screen is at NCBI or you're doing something else.
No, I have opened the NCBI screen only, ma'am.
Ha, okay, fine. So I told you you make the folder of Bioinformatics Workshop.
I did make it, ma'am. I made it.
You made it. Perfect. Now select any protein of your choice from the drop down, Nandini. Change
Okay.
the drop down, not here, in the all databases drop down. Ha, yes. Change it. Ha. To protein, because the protein sequences are small, I'm doing that. The DNA will be very big. Give any name of your choice of protein. Okay. Okay. Click on search. Yes. Okay. So the first one is some other species.
They're all insulin-like growth factor they are showing. And
Ha.
first, yeah.
Ha. You can take any one for practice right now. There is no problem.
Okay, okay.
Ha.
So I take this 4 number.
Oh, yes. My purpose is to teach you how to download the FASTA. You can do it for any protein.
Okay. Ma'am, what exactly is this faster?
FASTA sequence is C. This amino acid sequence, how they are taken from the wet lab by sequencing methods. And that data is deposited in the databases. But each database will have its own standard way of writing the files. Any
Hmm.
format is not accepted by the tools also. Any random sequence format, just the order is not acceptable. So it has to fulfill the criteria of FASTA. FASTA, if you see, it always starts with greater than symbol, then it will be written accession number, then the species number, and then the actual sequence. So if you miss out any of this, even that greater than symbol, tool will throw error. Okay. So this is your FASTA sequence, this greater than symbol thing. Hmm? This entire is your FASTA sequence. Yes.
Till this.
Till the last one, till the last residue of the sequence.
Up till
Yes.
here.
Yeah. You can make a word pad, copy paste it there, and name that file as a FASTA.
I saved it, ma'am, as faster.
Okay, fine. Then go to another database, Uniprot. Type in the Google Chrome as Uniprot. Huh? Huh? Nice. Now select any protein. You can select insulin, which is there in the examples also. Okay. Click on search button. It is asking you because you are opening for the first time. Would you like to see the information in card or table? Table is always good one. Select table. Perfect. Now select the first protein. I had done for the same. Click on the file. Click on the accession number. Yes. Perfect. Now there is download legend. Can you see, Nandini? Besides tools. Perfect. Format. Change it to FASTA canonical. Perfect. No, you just have to change that. Don't go into all this data set entry. Only change, this is your FASTA. Again, copy paste into the same file also you can do. Otherwise, make two
If
separate
they
files,
both,
one.
if they both are the result of um, same thing, I mean, like if they both are,
FASTA. Yeah.
uh,
You can
then
keep it in the same word. Yeah.
then they should be same.
Yeah, they are same.
Okay. Then why at two different places?
Why? Because Uniprot, I told you, is easier and more fast and segregated. Only one protein at one time will be included in the database. NCBI is like it is divided. One file will be for partial, one file will be for stretches of the sequences, and it is too much of information. So filtering time wise also becomes difficult. So we go for the quick as UniProt because it is only confined for protein. NCBI will be having nucleotide, genes, DNA, RNA, protein. So it is time taking. That's all. That is the reason.
Okay. Okay.
It's your comfort. I prefer to work fast on UniProt, so I do that. If you like NCBI, you can do that. No problem.
Okay.
Okay. Yeah. Now done with this, then we will go to PDB.
What is it saying? Please move this away.
But I can't see.
Just okay, maybe now it will. Please move this window away from the shared application.
ये वाला window. Anyways,
इसको
you have
यहाँ
saved it. Saved it.
yes, ma'am.
Then close. No problem. Then open PDB.
Just a minute, ma'am. This is copy. What if I write it for comparison? Here, FASTA NCBI,
Huh.
then FASTA UniProt.
No problem. Yeah.
So you said, ma'am, the difference was,
Just
can
the
you please
retrieval.
repeat? Yeah, in case of Uniprot, it is more systematic, precise. And in NCBI, lot more informations are there,
No, no,
right?
not in the sequence. Sequence will be same. The way of retrieval in NCBI for the same protein, you have all information in the form of dumped. So you have to filter key which is most relevant file. This is not the case in Uniprot because scientists manually work on it. Only one entry is allowed for one protein. So if you type human insulin, it will be very specific, only the human protein sequence. So it is faster and well segregated. That is the only
Okay.
difference. Last result, faster sequence has to be same.
You see, ma'am, in this case, it is different, the sequence.
No, no, it can't be different. If it is some subspecies or the partial or whole, then it will be different. That's why I told you in NCBI, it can be sometimes partial. It can be only chain A. It can be only chain B. Title will mention that. Here in Uniprot, you had a title where it was written complete 110 amino acid sequence. So it has to be same. If the same title, if you find from NCBI, it has to match. If there is any precursor, any other chain, then the sequence will differ. So what you are giving as a query is also important. Do not give it casually. Understood?
Okay, okay, okay,
Sequence has to be similar, no doubt about it. Hmm?
okay.
Okay, now show me the next one, PDB.
In case of PDB, we have just the protein. Uniprot, we have just the protein. While in case of NCBI, it can be protein or uh,
Exactly. Nucleotide, gene, everything.
yeah, yeah.
So, yeah. In case of Uniprot, it is only the protein sequence. In case of
Okay,
PDB, it is only the protein structure.
okay. So should I go for, I mean, after
PDB.
searching,
Huh?
yeah, I did open the PDB, enter
I
search
can't
term in,
see. I think your screen is frozen back. Are you doing it for PDB? I can't see. Huh.
now is it visible, ma'am?
Just a minute. Huh? Yeah, perfect. Now in that window, type your query protein.
Advanced
Ha,
search.
ha, ha, ha. Any protein of your choice. If you know the code, you give the code. If you want to give the name, name. Hmm. Again, there are so many sub entries. So according to
Yes.
the information deposition, it will make different file. Out of
I
that, the
mean,
most relevant one you have to select.
I mean, because here the entry are done by different scientists working on the same thing, if they are
Ha.
accepted. It's same for Uniprot NCBI as well.
Yes, yes, yes.
So what we see is what has been, I mean, researched upon by the scientist at all the three stages. It means the three are like the repository of the result of the research on the given protein, right?
Correct, correct, correct. Research papers. Yes. It's all
Okay.
based on data mining.
So can it be, there'll be ambiguity also, or they do not allow ambiguity when they are totally satisfied, then also they put it.
Exactly. Their procedure is so stringent that it is always validated. But still, when you do this and you are into it, you will understand gradually. Now that I'm working into it thoroughly, I understand there are some ambiguities in the PDB files, not in the sequences. Why?
Ma'am, you know,
Because
in
PD,
2015, I had done research on TLR6 gene, my
huh,
PhD work in case of swine, but I was not able to put the sequence on any of these, I mean, for verification to any of these forums. I mean, because it is said if you are able to upload the sequence and they accept it, then you get some marks. Right, right.
Ha ha ha.
But I was not because I did not know the procedure to upload the. I
Acha.
don't know how valid it will be now.
Hmm. Okay,
So
okay.
this is a human insulin protein.
Yeah.
Okay, so out of it,
Hmm.
suppose I take the first one.
Hmm.
So it is opening.
But again, you see, there is a subdivision. It is type 1 and that too receptor protein. So that's
Yes,
fine.
yes.
Now our purpose is to take the understand downloading the PDB file. It's okay. But when your
Okay,
objective is clear that suppose you want to work on corona protein, only a spike protein, then you
okay,
have to take that PDB file only. You can't just type corona and take the membrane and do the spike and then you want to perform docking on spike. Then the results will be wrong. So
okay,
target identification is also equally important. Hmm?
okay.
Now what you'll do, click on that code.
This.
6 kj. Code. Yeah. Then on the right hand side, you can see the bright download file option. Yes. Out of that, you will select legacy PDB format. No, no, no. This you have done wrong. This is very commonly done mistake by newcomers. Don't worry. Legacy PDB format. Yes.
Okay, okay,
Okay. You have to select only this format. Now you have to transfer it from this folder into the working directory. And as I mentioned, you can open and see the Word files of FASTA, but you cannot open it without the support of visualization software. So
okay,
that we will install tomorrow. We have to install it on your system, Swiss PDB Viewer and Rasmol. Okay.
okay, ma'am.
Yeah.
These all actually they taught me, ma'am, in Tanuvas. But the fact is that there were many things like it was introductory for the first time, or it was on the topic, and many topics were there. Like it was introductory for all topics. So at that time, I felt I have understood. I will be remembering it for lifetime. But now I have forgotten everything. So
Yeah,
maybe I could
I
have
tell
given
you now, bioinformatics is a very volatile subject. If you are not in practice, you will forget everything.
exactly, ma'am. That is what I felt.
Yeah, it
So
is like that.
this, ma'am. This is the thing.
Yeah. So why? But again, when you start getting command on it, when you practice it, again you will gain that confidence.
Okay, okay.
Yeah. Now you have stamina still. Should I cover one more tool?
Ma'am, yeah, I have. Actually, I'm not able to perhaps share it. What I got, I want to show to you.
Hmm.
I opened the file I downloaded from
Huh.
PDB.
Yeah. So it will open in the form of notepad, but you can't make really sense of it. When you have visualization software, you can watch it in 3 dimension beautifully. Okay.
Okay,
Yeah, this is information of the residues in XYZ coordinate. And when you take this file with .pdb extension into visualization software, it looks like a beautiful protein.
okay.
Have you seen it before in the
Y-
workshop?
Yes, ma'am. They
Aha.
helped us download that Rasmol and
Ha, so
then
I
Swiss
thought
dot.
if you are enthusiastic, I will quickly show it on my system if you don't have softwares installed on your system. But if you already seen, then that's fine.
No, no, please show me, ma'am. Kindly show
I'll
me.
show
It's been
you.
long, ma'am. Yeah,
Okay.
sure, ma'am.
We will do it on your system tomorrow. Don't worry. But for your understanding today, I'll show you.
Okay, ma'am.
So I have multiple softwares because I work a lot on all these things. So there is one Chimera, there is one Discovery Studio, there is one Swiss PDB viewer. So tomorrow we are going to use Raswin and Swiss PDB viewer. So I'll teach you on that. So it looks beautiful on the Raswin also.
They showed us Chimera, Swiss
Ha
PDB,
ha ha
Swiss PDB viewer,
ha
and
ha ha
Rasmol. Three they had discussed as far
ha
as
ha
I remember.
ha. So this kind of visualization, right? So you can see it in different displays also. This is wireframe backbone
And you
stick.
can share the screen. I
I
mean,
am not sharing. Okay.
no. Or maybe the screen is frozen because I can just see BDG Life Sciences
Ah,
Private.
acha, acha. I will just check what is the problem. Now can you see?
Yes, ma'am. Yes, ma'am.
Ah. So this kind of beautiful protein, you can see this is RASMol. Similarly, you can open the PDB file in PyMol in Discovery Studio and then make analysis. So there, this also I take in bit details for some programs. I will try to teach you more and more things whichever is possible in the schedule. So in the PyMol, we run it command based also. We run it GUI based also. If you want to see same molecule in Discovery Studio, let me show you, because this is a most commonly used software for the analysis also. And when you're doing docking, you have to be really good in operating DSV. So we go to file, open any PDB file you can open, set it, look like this. Now here you can manipulate, you are going to do in your advanced sessions many things on this, removing water, adding charges and docking, everything. Okay, cleaning the ligand for protein during MD simulation. So it has a lot of very important features during docking also, to see the 2D diagram of the interaction between protein and ligand. It is very important. So quickly I'll show you. Achha. Now we'll have to define the ligand. It is a bit process. When we are doing this visualization, we will do all the exercises for this. Okay. Now, yes. Tell me, Nandini, shall we proceed with next tool?
Okay, ma'am. Okay, ma'am.
Okay, na? Otherwise, if you're tired, tomorrow we can do. No problem. We can cover up.
No, ma'am. One tool you can
One
cover,
tool.
then I will,
Okay, out
so
of
that I
three,
will practice.
out of three, one we will cover. Okay,
Yeah,
so
yeah,
that
ma'am.
it
From
is not
tomorrow,
much.
I will be on time so that it's
Yeah.
not it.
Okay, no problem. So now the first tool for the analysis of the protein properties we are going to see, that is known as broad scale analysis. Now, what is a broad scale analysis? Broad scale allows to compute and represent in the form of 2D plot the profile produced by any amino acid scale on a selected protein. So try to understand what does this sentence mean. We are going to take the 2D plot of a protein. So we have a sequence of amino acid in the form of protein. We are going to move across horizontally this sequence, and we are going to focus each time on one part of the sequence. So that is called as a window. At a time, suppose I am starting from first amino acid to 50 amino acid. So I will first think about 5 amino acids in the window. Now I have a data of the propensity values or the scale values which are assigned to each amino acid property. So suppose I am focusing on hydrophobicity prot scale. So I will have data of hydrophobicity values for each amino acid, which are known as scales. And these we are plotting and joining in the form of 2D plot. That is called as a plot scale. Now, this technique is called as a sliding window technique. Why I call it as a sliding window? It is just like a sliders of your window. You first focus on the first, draw the peak, shift to right by one amino acid. So your next window will come into picture. Then again you draw a peak. Then the next, till you reach the end. So this is the simple logic. Now, operating is very simple. This all calculations, almost all the BI tools, all the applications are running at the back end. You just have to run the query. Tool will not bother you at all. They are so nice that they will not trouble you. It's all mostly the data mining based, AI based. So you just have to give your query and do the thing. So suppose here I have a box, uniprot number, accession number, which I explained you can enter, or else we have just learned how to paste the sequence, faster sequence. You can do that, and you have 20 scales or properties. Out of that, you'll select anyone. Suppose I'm interested about the hydrophobicity. So I'll click on hydrophobicity, kite and Doolittle, and click. So I get this kind of beautiful graph in the form of ECG. How to read this graph? So this is for insulin. You can do it for anyone. On the x-axis, you have positions. On the y-axis, you have scores. So these positions you are going to relate with the peaks. Always remember top 3 peaks you will see. So for me, this is the topmost peak. If you extrapolate it on the x-axis, it is the 10th residue. So 10th residue, then you see this is 38 residue, and then you see this is 98 residue, which are showing topmost peaks. So how to interpret that? Hydrophobicity is maximum at these 3 points. And you can see one deep trough over here, which is around 58 position. So that is most hydrophilic region. This is the simplest way to know the highlighting features of the protein. And if you see maximum hydrophobic regions, that means your protein is likely to be a receptor protein. Then you validate it with one more software known as TMHMM. That we will do tomorrow. You perform this and then we will stop for today. I'll show you how are you going to access this software. So simply go to Google Chrome, type prot scale. It runs on XPASI server. So I click on prot scale. Again, here you can copy paste the sequence, or else you can give accession number. Let's say I'm taking this sequence because it was ready in my hand. You can take anything. Now, by default, it is hydrophobicity because it is most commonly used. You can select any property which you are interested in. Then I click on submit button. That's all. Very nice and quick. Yeah, only the color they have changed. In my PPT, it was the old slide. So now they have recently changed it to golden color. So, but the interpretation is same. Can you see? This one is your 10th. This one is your 38. This one is your 98. So what you are supposed to give me as a result would be this image. Along with that, you will write an interpretation as top 3 peaks are observed at 10th, 38th, and 90th positions. So, hydrophobicity for insulin is concentrated at these 3 points, making it a hydrophobic protein. Am I clear on this?
Yes, madam. Yes, madam.
Very good. Now you share your screen. We will do this and we will stop. Hmm?
Okay, ma'am. You will have to again. I'm sending the request, ma'am, to share the screen.
Yeah, yeah. I've allowed you, I think.
Okay. So go to PDB,
Yeah.
select the sequence.
No, 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 not PDB. You have to go to UniProt. We don't require structure, we require sequence. Share your screen, I will help you.
Mm. Ma'am, am I able to share now? I think you can see.
Now it has started.
So you told Uniprot, right?
Ha,
Ma'am, what did you say the reason, Uniprot?
because UniProt is having sequence, PDB is having structures. We need sequence for UniProt. For prot scale, we need sequence, not the structure.
Okay, then only we will be able to deduce which amino acid is hydrophobic, which is hydrophilic.
Right. Because this is primary structure analysis. When you have to do tertiary structure analysis, we will take PDB file. When you have to
So
do primary and secondary, sequence.
Uniprot gives us the
Sequence.
sequence. PDB
Yeah,
gives us the structure.
correct.
And NCBI?
Sequence. PDB is
Okay.
the only database which is used for sequence. Sorry,
PDB is
structure,
only
structure,
structure.
structure,
Structure.
structure. Yeah.
So what is the added use of a structure when we already have sequence with Uniprot and NCBI?
No, it makes a lot of difference because sequence is only two-dimensional order. 3D is how these amino acids are arranged in 3 dimensions. So for
Okay, and that
performing
in vivo
position,
interaction in biological
yeah.
system.
All docking MD will be entirely based on how they are arranged in 3D. So PDB is very important.
Okay.
And when your PDB is not available in the database with the sequence, there are now AI-based softwares which makes the models also for novel
Okay.
proteins.
What did you say last line, ma'am?
When now I get such queries or clients where they don't have their common PDB proteins in the database. In such cases, suppose they have worked recently in the lab and they have come up with very novel protein sequence. So in
Okay.
that case, they don't have the database which will give their protein. So we have to build the model on the basis of this sequence information. So for that, there are some new AI-based softwares are introduced like AlphaFold 3.
Okay.
But
Okay.
anyways, you have to convert it into PDB before you proceed for docking.
Okay. 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 कौन सा ligand कहाँ attach करेगा? It is possible to know only with tertiary structure, which
Tertiary
is possible.
structure.
Okay. So I come here, ma'am, and then I have to go for what was the thing you said?
Download pasta canonical. Copy this. Open your prot scale tool. Type in the Google Chrome as prot scale. Just scroll down. There will be Yeah, very good. Paste the sequence. Very good. Ha, it is already selected. So just go and submit. Perfect. Perfect. Very good. Take the screenshot because saving the file in GIF format and all and converting becomes difficult. No need. Take a proper screenshot. Clear. And save it and name the file as plot scale output. I want you to write interpretation also below the image. What will be your interpretation, Nandini? Tell me.
At this is most hydrophobic at about position 10, the amino acid at position 10, and least hydrophobic, yani most hydrophilic at about position say 40 हो गया 50 हो गया 55 55.
Good. And first the three peaks you always see. So first will be 10th, next is your 38 and last is your 98.
Okay, ma'am.
Huh.
First is our 10.
Huh?
Here. First is our 10.
Yeah,
Next is our peaks. Next is 38. Last is
correct. 98.
90.
Okay. Yeah.
Okay, the three most hydrophobic peaks are shown
Yes.
at. Okay.
Okay.
So, just for a knowledge, what is the use of this hydrophobic?
So, hydrophobic protein nature means if you are completely new, you don't know the character of the protein. First thing that you have to check if it is water loving or water hating. If it is hydrophobic, then it might work as a receptor protein. So then you will follow those steps which will confirm whether it is receptor or secretory. So next to this, tomorrow we will do TMHMM. In TMHMM, we will load the protein and check if it has transmembrane helices. If it has transmembrane helices, then that protein will be receptor. So like
Okay,
that, by shortlisting the characteristic, you can reach to more specific information about the protein. Once you're
okay,
sure about this, we will also run one prot param tool, which will give the basic properties of the protein. Once you are
okay,
done this, then the protein will be taken for secondary structure prediction. Once you know the basic properties, then you will go for secondary structure. Then you will go for its visualization in 3 dimensions. So like that, your application is moving towards more specificity from the general.
understood, ma'am.
Okay, very good.
Mhm.
Okay, then we can stop for today.
Okay, ma'am. Thank you so
So
much, ma'am.
my pleasure, dear. So you will
Mhm.
write the interpretation, okay,
Okay,
that
ma'am.
you can do afterwards as a homework. Okay.
Okay, ma'am.
Fine. So tomorrow
Okay,
we will meet and tomorrow we will proceed with the prot-param, TMHMM. Then I will also cover secondary structure prediction. And if the time permits, I'm also planning to cover Swiss PDB. Let us see how it
sure,
goes. Okay.
sure, ma'am.
And today's recording and today's notes of the topics and the steps, everything I will share now on the group. Recording might take some time because it takes time to load and then my technical team will load it. But notes and flowchart I will share immediately now. Okay.
Okay, ma'am. Okay, ma'am.
All right,
Uh,
dear.
you are Sharvari Kulkarni, ma'am, right?
Sharvari, yeah.
Okay. I'm
Okay.
saving your number, ma'am. Okay.
Sure, no problem. Yes, anytime, anytime.
Thank you, ma'am. Okay.
Okay, dear. हाँ, चलो take
Okay,
care. Take
bye.
rest
Good
now.
night.
You have had hectic day.
Yeah, it was a same with you. I understand, ma'am. I
Yeah,
understand.
it was tough to manage her with maths. But now she is happy because I got engaged in sessions, so she had relaxed time.
Okay. Yeah, yes, sir.
Okay, dear.
Okay. Good night,
Bye
ma'am.
bye.
Okay.
Bye, good night.